Good morning. Today we're going to talk about cancer biology, and I'm going to be following along pretty closely to the Chapter 12 uh, PowerPoints. I'm not going to talk about the epidemiology or a little bit about the childhood cancers, but I thought this was the chapter that needed the most explanation. Cancer is derived from the Greek word for crab, and it's another name for malignant tumor, so that when they say you have cancer, they're not usually referring to benign. Cancer is not a tumor, it's an abnormal growth resulting from uncontrolled proliferation. And basically, it's cell growth and division out of control. And it's also referred to as a neoplasm, which means new growth. You have in your PowerPoint there on page 1 the difference between benign and malignant. Basically, this should be a review for you. Benign is a confined growth, it's slow going, it's well defined, it's not invaded the blood and the lymph and surrounding tissues. It's well differentiated. In other words, the skin cells look like skin cells and the muscle cells look like muscle cells. They don't, all the cells don't look alike. Like all the cancer cells that are undifferentiated or dedifferentiated look alike. They're all cancerous. It has a low division index and it doesn't not metastasize, whereas malignant is just the opposite. It's dividing rapidly. It goes unchecked in terms of growth. It's not encapsulated. It's invaded the surrounding tissues. It's poorly differentiated and what we call anaplastic, and high mitotic index. It divides very quickly and often, and it can spread and has metastasized. Malignant tumors, in terms of the nomenclature, are named for the tissues for which they arise. So just some examples: sarcoma comes from connective tissue tumors, lymphomas from lymphatic tumors, leukemias from blood-forming cells. A carcinoma in situ means that it's a pre-invasive epithelial malignant tumor of glandular or squamous origin. It hasn't broken through the basement membrane or invaded into the surrounding tissues. It's not malignant. It's stable. It can progress to a malignant cancer, but it can also regress and disappear. Tumor markers. Certainly we use tumor markers in terms of breast cancer, prostate cancer, and basically they're substances that are produced both by benign and malignant cancer. Like, for instance, with the PSA, which is a tumor marker, tumor markers are found in the tumor cells in the blood, spinal fluid, or urine. And they can be hormones, enzymes, genes, antigens, or antibodies. In terms of a tumor marker, we use it to tell us a lot of times in terms of the successfulness of the treatment. If the treatment is successful with prostate cancer, the PSA will go down. But it also, you can't use it as gold standard for diagnosis because it can be elevated with many other different reasons other than a malignant tumor. And tumor markers are used to screen people at high risk for cancers. And like I said, if they're successfully treated, they can go down. And later, if they come back up, it might mean a recurrence of that cancer. It goes into the biology, that particular chapter goes into the biology of cancer cells. And there's some very important definitions. Transformation is the process where the normal cell becomes a cancer cell. And when that happens, a cancer cell is dividing out of control. It divides without the usual stop mechanisms. It's autonomous. So for instance, it independent from the normal cellular controls. When it divides, usually the cells, when they touch the neighboring cells, they stop dividing. But in the case of cancer cells, they just keep on, no matter how crowded it is. They also are anchor independent. They're not stable in terms of a tissue. Um, the cancer doesn't mind that it's floating around. It doesn't have a home or a solid base of support. It's immortal. It has an unlimited lifespan, and this goes back to the fact that you have the telomeres at the very end, the end of the genes, and normally those get smaller and smaller and disintegrate so that the cell dies. In the case of a cancer cell, that doesn't happen, and so that the cancer cell lives on and on. The enzyme that usually makes that happen is interfered with. Anaplasia occurs, so you have different sizes, different shapes, the absence, like I said, of differentiation, or it's de-differentiated. The skin cells don't look like skin cells. They're all alike. They're all cancerous. They're not differentiated in terms of different types of cells, and they all look the same. There's a loss of organization, specialized functions, nuclear size increases. There's no, it lacks a uniform shape and size, and it's variable, and we call that pleomorphic. The metabolism is also changed. Basically, the cancer cell is a parasite, and it goes in and takes advantage of the oxygen and the nutrients. And most of the time, the cell needs oxygen and it needs a stable environment. The cancer cell can grow in hypoxic conditions and acidic environment. And it also it takes the nutrients from the tissues themselves, and it performs glycolysis, even in the presence of oxygen in the tissues. And it allows lactate and it's metabolized to be used for production of lipids and other building blocks needed for cell growth, and thereby is constantly growing and has the basis and the building blocks for which to grow. Cancer cells, like I said, are self-renew themselves and create their, like intestinal cells, bone marrow cells repopulate in a certain period of time, whereas cancer cells are constantly dividing. Cancer cells, cancer in itself is usually, it can occur in children, and other age groups, but basically is a disease of aging. Their expansion occurs, like he said, and multiple mutations can occur before the cancer appears. Usually the cancer needs 2 or more hits. Like for instance, an individual who is at risk genetically for melanoma would need many different episodes of getting sunburned and exposed to ultraviolet rays, and have many different genetic hits, and in terms of mutations to occur for the full-blown cancer. It's a stepwise accumulation of different alterations in specific genes. So it needs exposure to many causes: chronic inflammation, repeated infections, smoking, obesity, alcoholism. So usually there's more than one factor, or it occurs repeatedly or chronically over a period of time. Basically, cancer-causing mutations activate these growth promoting pathways. They block anti-growth signals. Mutations, they prevent apoptosis. So in other words, there's no time span on the cells themselves. They just continue to live on and divide. They turn on the telomerase, which is a specialized enzyme active only in stem cells and maintains the telomeres. So the telomeres do not shorten, and they continue. The cell doesn't disintegrate, and it continues to divide. It also starts its own blood vessel growth. angiogenesis, and it's stimulated by factors like VEGF or PDGF, depending on the tissues, among others. And it allows tissue invasion and metastasize in different tissues, like through the blood, through the lymph, or in surrounding tissues by invasion. Other important definitions is the definition for oncogene, a mutant gene that increases in its cell division and proliferation and directs its own protein synthesis. Tumor suppressor genes is an encoded proteins that in the normal state negatively regulate proliferation, but they're anti-oncogenes. They prevent cancers from occurring, but in usually cancer, they're turned off. Proto-oncogenes are normal non-mutant genes that are coded for cellular growth as a growth factor or growth factor receptor. Or one example is RAS, which switches on other proteins involved in cellular growth and proliferation. There's many keys to cancer development. Sometimes there's mutations in one or more nucleotide base pairs. Like I said, the RAS gene converts a regulated proto-oncogene to an unregulated oncogene with accelerated growth. You can have chromosome translocations. In other words, a piece of one chromosome is transferred to another part of the chromosome. Gene amplification, which is a small piece of the chromosome that is duplicated over and over again. So you have thousands of copies in this one abnormal gene, and that can lead to abnormal protein and protein production and cell growth. Other causes of mutation is the inactivation, like I said, of the tumor suppressor gene. This is the anti-oncogene, and it allows unregulated cellular growth. It takes 2 hits to activate 2 allies of this tumor suppressor gene. So normally the tumor suppressor gene slows or stops the cell growth, but in this case it doesn't happen. There's a loss of ability to be different. Both chromosome copies of the gene are inactivated. It can unmask recessive genes so that 2 different copies become the same, and that it keeps replicating the mutation. Genes silencing, where whole regions of chromosomes are shut off while some regions remain active. Does not require mutations or changes in the DNA sequence. And this gene silencing is a major cause of cancer. Chemical changes in the environment, altered expressions of the genes produce different types of tissues. So the expression is different than normal. So the skin cell doesn't look like a skin cell. And in various tissues, it might even revert to a stem cell, which is a rapidly dividing tissue. And hopefully this will allow us in the future some new chemotherapeutic drugs that can reactivate these silencing genes. Other things that we see is changes on the genes are not necessarily in a single area, but networks of genes, and in many, many different genes, which causes a difficulty and changes in proteins, DNA, and the expression and the function of the cell itself. Guardians of the genome and how mutations occur are largely responsible from the caretaker genes. And their caretaker genes are responsible for the maintenance of the integrity of the genome. They're encoded proteins that are involved in repairing any damaged DNA that occurs with errors in replication